My name is Sophia, and today we'll be talking about muscles. Let's start off by exploring different types of muscles. Cardiac muscle is found in the heart. It is striated and involuntary, pumping blood throughout the cardiovascular system. Skeletal muscle is attached to bone with tendons. It is striated and voluntary, allowing for movement. Smooth muscle is involuntary and found in hollow internal organs, such as the stomach. Let's take a look at a few muscle tissue samples extracted from a rat dissection. Taking a closer look, you can notice the striations in skeletal and cardiac samples, while the smooth muscle sample extracted from the intestine has a smoother structure. Differentiating between cardiac and skeletal muscle can sometimes be difficult. Skeletal and cardiac muscle may look similar, but one key aspect that sets them apart are the intercalated discs connecting the cells of cardiac muscle to one another. Smooth muscle does not have any striations present, and you can clearly see it is an outlier. The voluntary and involuntary muscle contraction experiment we performed in class tested the blinking reflex. Most of the time, our test subjects blinked as a reflex, which is an involuntary muscle contraction. Muscle contractions happen due to signals being sent by motor neurons. Simply put, the signal is received by the neuron and is sent down the axon. The electrical impulse travels with the help of sodium-potassium pumps located along the axon. Sodium and potassium ions move in and out of the axon, moving the signal. When the action potential reaches the end of the axon terminal, neurotransmitter acetylcholine is released. This process eventually leads to a muscle contraction. How do you think salt would affect the muscle contraction? After testing and measuring muscle contractions in 4 different solutions, we can conclude that ATP and salt are important for facilitating muscle contractions. To understand muscle structure, we can use a mandarin as an analogy. The outer skin is the epimysium, covering the fascicles that are bunched up on the inside. They are divided by the perimysium and hold muscle fibers. Fibers translate electrical impulses into mechanical responses through excitation-contraction coupling. When the electrical impulse reaches the ends of an axon terminal, acetylcholine is released from the synaptic vesicle. It is then diffused into the synaptic cleft and attaches to ACH receptors on the sarcolemma at the neuromuscular junction. This causes the muscle fibers to become more positively charged, which generates action potential that travels through the sarcolemma and along the T-tubules. As it travels, calcium is released from the sarcoplasmic reticulum. Calcium then makes its way to the sarcomeres, where it binds to troponin on the actin filaments. This signals tropomyosin to move out of the myosin binding sites. Energized myosin heads with an attached ADP and phosphate group attached to the open binding sites, forming an actin-myosin cross-bridge. The myosin head then rotates towards the sarcomere's center, releasing ADP and phosphate, which slides the actin filaments past the thick filaments, resulting in the power stroke. The actin-myosin cross-bridge remains intact until an ATP molecule binds to the myosin head. After it detaches, the myosin detaches from the actin.